We're going to take a look at our last two organic molecules, proteins and nucleic acids. Proteins have a lot of different functions because they have a lot of different shapes. So in the cell, proteins can serve as enzymes. They can also be used to transport things across the cell membrane. They can be used for movement because they make up the flagella and cilia. Some bacterial toxins are made out of proteins, and also they can be used for structure as part of the cytoskeleton in cells. Polypeptides are polymers that can be made from up to 20 different amino acids. And remember, the amino acids are the monomers or subunits or building blocks of the polymers. And the amino acids will be joined together by peptide bonds to make up the polypeptides. So let's just take a look at the structure of the amino acid or the monomer, the building block of the proteins. So here's a picture of an amino acid. So they are made up of a central carbon in the middle. And off of that central carbon, we've got an amino group which has a nitrogen in it. And then on the other end, we've got a carboxyl group. And then on the other side, we've got an R group. And the R group or side group is what differs between the 20 different amino acids. And amino acids are going to be joined together in a dehydration synthesis reaction. We just remove the water molecule and then just form a covalent bond between the amino acids, and that covalent bond is called a peptide bond. So peptide bonds will just join the two amino acids together. Okay, so there are 4 levels of protein structure, and this would be important to remember. So the primary structure would just be the linear sequence of amino acids, so just what order the amino acids are in. And the primary structure is just going to be held together by peptide bonds between the amino acids. The secondary structure, we just take that primary structure and then we just fold it and form either an alpha helix or a beta pleated sheet. And those will be held together by hydrogen bonds. And then the tertiary structure, we just take the alpha helices and beta pleated sheets and fold them even more. And the tertiary structure would just be the overall 3D shape of the polypeptide chain. And these are going to be held together by different chemical bonds between the R groups or side chains on the amino acids. And then the quaternary structure, we're only going to have if we have 2 or more polypeptide chains. So if you only have one polypeptide chain, you would stop at the tertiary structure, but if you have 2 or more polypeptide chains, then you would have a quaternary structure. So here's a nice picture just showing the 4 levels of protein structure. We've got the primary level of protein structure. This is just the linear sequence of the amino acids, so just what order the amino acids are in. And this is just held together by the peptide bonds between the amino acids. And then we're just going to fold this primary structure up to get our secondary level of protein structure. So we can either get an alpha helix, which is just kind of a coiled structure, or a beta pleated sheet, which is kind of an accordion shape. And these 2 structures are going to be held together by hydrogen bonds. And then we would just fold these up to form the tertiary structure. And the tertiary structure would just be the overall 3D shape of the polypeptide chain. And this would just be held together by the different interactions, different chemical bonds between the R groups on the amino acids. And then if you have 2 or more polypeptide chains held together, like this one has 2 polypeptide chains held together, then you would have a quaternary structure. But if you just only have 1 polypeptide chain, then you would just stop at the tertiary structure. Okay, and then what's really important to remember is that the shape of the protein is going to determine its function. So shape determines function. That's a really important concept to remember. Okay, proteins can denature or unfold and lose their three-dimensional shape when they're exposed to heat, or maybe the pH isn't right, maybe it's too acidic or too basic, or maybe they're exposed to salty solutions or different solvents, anything like that. And the only thing that is left, so if you look at this three-dimensional shape over here, and maybe we heat this protein up and denature it, the only level of protein structure that's left would just be the primary structure. So the only part that's left is just the linear sequence of the amino acids held together by the peptide bonds. So what happens to the protein function is that it no longer works because you've lost that three-dimensional shape, and so it's no longer going to do the job that it was doing. Right, our fourth and final organic molecule we're going to talk about are nucleic acids, and nucleic acids are polymers that are made up of nucleotides that are joined together. So nucleotides are the monomer. And there are 2 different types of nucleic acids. We have DNA, which is deoxyribonucleic acid, and RNA, which is ribonucleic acid. And the functions of DNA and RNA: DNA will transmit the hereditary information from one generation to the next, and then the genes in the DNA will code for proteins, and messenger RNA will help make the proteins. Nucleotides are made up of 3 things, so this would be important to know. So they are made up of a nitrogenous base, a pentose or 5-carbon sugar, and a phosphate group. Okay, so if we look on our picture over here, here's a nucleotide, and the nitrogenous base has nitrogens in it, so that's how it got its name. And the nitrogenous base can either be a purine or a pyrimidine. And all you need to remember about this is the nitrogenous base is just going to be the A, C, G, T, or U. So that would be the nitrogenous base. And then the pentose or 5-carbon sugar will either be a ribose for RNA or a deoxyribose for DNA. So the only difference is that the deoxyribose has a missing oxygen. And then there's also a phosphate group. So I would just know what a nucleotide looks like, and it's got those 3 parts. Okay, these are what the nitrogenous bases look like for the nucleotides. I'm not going to ask you which ones are pyrimidines and which ones are purines. I will not ask you that. But here's what they look like. So just you've heard the names and you realize that pyrimidines and purines are nitrogenous bases and they're part of the nucleotides, and you just realize that they're the letters, the C's, T's, U's, A's, and G's. That's all I want you to know about those. Okay, and with the structure of DNA, remember that we've got 2 strands, and hydrogen bonds are going to hold together the 2 strands in the middle here between the nitrogenous bases, and it forms this double helix. And the rungs of the ladder are formed by the nitrogenous bases. And then the outside of the ladder, that you would hold on to as you're going up here. These are the sugar phosphate backbone. Okay, so the sugar phosphate backbone is on the outside, and the nitrogenous bases make up the rungs of the ladder. Okay, and then just remember the DNA contains the genes, and those will code for how to make the proteins and different types of RNA like transfer RNA and ribosomal RNA. And this is what's inherited and passed down to the daughter cells. Okay, and here's just a nice summary between the differences between DNA and RNA. So DNA is double-stranded where RNA is single-stranded. And then the sugar in DNA is deoxyribose, so there's a missing oxygen, and ribose is the sugar in RNA. And then in the nitrogenous bases, DNA has T's, whereas RNA has U's instead. And this is just a nice summary from your book that just summarizes the 4 organic molecules and their functions.